8th day after fertilization. See it in the diagram what change occurs. This is the trophoblast. This is the inner cell mass, and what happens, cells of the inner cell mass which are facing to the blastocele, they become cuboidal and they form the endoderm. This is the first germ layer. Soon after formation of endoderm, the other cells become columnar and form ectoderm. That is the second germ layer on 8th day after fertilization. See it, this is the trophoblast. This is ectoderm. And this is the endoderm. So these are the two germ layers are formed in this manner, and now there are two cavities. This cavity is known as primary yolk sac, and this cavity is known as amniotic cavity. Now see formation of third germ layer. Formation of third germ layer. Third germ layer is the mesoderm. This mesoderm is last to form on 16th day after fertilization. Formation of mesoderm is known as gastrulation. Now see how the third germ layer is formed. I am making two germ layers separately. This is the columnar cells of ectoderm. And inferior to it is the cuboidal cells. This is the endoderm. This stage is also known as the bilaminar germ disc. There is no cranial end, no tail end till this point of time. Now what changes occurs at either end, either this side or this side, these cuboidal cells, they change their shape and they becomes columnar. What I mean? On either end, this endoderm, they change their shape, they becomes columnar like this. This change is known as procaudal plate. It fixes the cranial end. Now it is fixed that head of the baby will form at this end. So the cranial end, it is decided by procaudal plate, which is formed by the endoderm. On which day? On 14th day after fertilization. Soon after the formation of procaudal plate at the other end, these columnar cells, they also change their shape and they becomes cuboidal in place of columnar, and this change is known as primitive streak. This is primitive streak. When does it appear? On 14th day after fertilization. What it forms? It forms the tail end. Now it has been decided that the cranial end and the tail end of the developing embryo. Now what next change occurs? Cells of the primitive streak, they starts dividing very rapidly. Cells of primitive streak starts dividing very rapidly. So heap of cells develop which start migrating between ectoderm and endoderm and form third germ layer that is known as mesoderm. See it, if this is the embryoblast, this is cranial end, this is tail end, C4 cranial, T4 tail. Cells at the tail end, they are rapidly dividing and they starts migrating between the ectoderm and endoderm. At every place they form the mesoderm. This is the ectoderm, this is endoderm, this is endoderm, and in between these two mesoderm appears. This is the mesoderm. So this is the third germ layer, mesoderm. Mesoderm. So in this manner there is the formation of 3 germ layers. Now one question, how many germ layers in the first week of development? Number of germ layers in first week. Answer is 0. Number of germ layers in the second week of development, there are 2 germ layers. Number of germ layers in third week of development, answer is 3. So these are the number of germ layers. Now what structure in our body develop from these 3 germ layers? First heading is mesoderm, which is last to develop, Mesoderm. What structures in our body are formed by mesoderm? Muscles are formed by mesoderm. Muscles means smooth muscle, cardiac muscle and skeletal muscle. All type of muscles are derivative of mesoderm except, what is the exception? Number 1, ciliary muscles. Number 2, pupillary muscles. Pupillary muscles are further divided into two parts, sphincter pupillae and dilator pupillae. Both are intrinsic muscles of the eye. They are intrinsic muscles of eye. So, all the muscles of the body, they are derivative of mesoderm except these three muscles, ciliary muscle, sphincter pupillae and dilator pupillae. They do not arise from mesoderm. Then from where are they coming? Ciliary muscles, it is the derivative of neural crest cells. C for ciliary, C for neural crest cells. In sphincter pupillae and dilator pupillae, they are coming from neural ectoderm.